It seems to be going, preparing to live stream. If somebody could be so kind of letting me know when we have confirmed live streaming. It is, we're okay with live streaming? Okay, so far so good. First step is in. So with that, I'm gonna start by sharing screen right away because that's the way I have organized the presentation. That way I can let you know what the plans are. So thank you so much for joining us. I want to keep an eye on my watch because we have an exciting full menu. And if you're joining us today for our 9th season, starting the 5th year of Fine, is because you must have received an announcement that looks more or less like the one that I'm showing you now. But it is a little bit misleading because we have so much more than my presentation and Sarah. So let me go straight to telling you how we are gonna organize our first hour today. First, I'm going to spend some time celebrating the Frontiers in Social Evolution seminar series that you have all made possible. We'll spend some time going over what we have accomplished all these years. Then I'll present maybe 20, 25 minutes on Titi and Almaki fathers, a lifelong perspective on the evolution of paternal care. Third, we'll have a treat. Sarah Blaffer Hrdy, I'll introduce her later. She will discuss Father Time, her new book, and the evolutionary puzzle of primate dads. And then we will wrap up the first hour with a raffle. We are very fortunate that 7 past speakers at Fine very kindly donated books they have authored. So you'll have a chance to enter the raffle and win a book signed by the speaker directly to you. You know that we're going to do this between 11 New York time and noon. And then at noon, if everything goes well, we should be ready to move on to our typical 1-hour discussion. Let me just make sure that, yes, we are recording. I think I'm all set. I'm not going to keep, if somebody sees something in the chat that I should be made aware of, please let me know because I do not keep my chatting open. I need to focus on what I'm going to share with you. Fine. So I want to give credit and I want to thank my colleagues at Fine, Karsten, Lauren, and Adriana. We have been running the program now for 4 years together. It is fair to say that it was Karsten's and Lauren's idea, and I joined them then, and then Adriana joined us as well. And we couldn't have done this without the help of some of our younger scholars, Facundo Fernandez-Duque, Jingyu Q, Madison Roberts, and Logan Platt have been very, very helpful. They are the ones doing so much behind the scenes that makes this series possible. So thanks to all of you. What have we accomplished? Let me just, there you go. When we started, I'm having trouble with my mouse. A focus of Fine and something to which I'm going to speak today is really comparative studies. But one of the reasons that I was going back to some emails with Karsten back in 2019 that really got us talking is a shared interest in natural history and data quality as they inform comparative studies primarily on behavioral ecology and social evolution. And we have attended to this interest of ours. You can see there a collection of talks and panels that addressed the issue of statistics, research design, data quality. We have also have panels, and we look forward to hearing from you on ideas, where we invited a number of colleagues to summarize their views on different topics. For example, editorial matters. We had one run by Latin American students. We had one on long-term field studies, and one on animal behavior and conservation. Lauren led a panel that summarized the work that we had done on teaching within Fine. And you can learn a lot about everything we've done for teaching, and I think you can benefit from it. If you go to our website, one of the things that we published a summary in ethology where you can get some ideas on how to use Fine to teach animal behavior. You can find a reading list, you can have teaching slides, And at the bottom left of the slide, you're just seeing 3 samples of quick find videos. These are 3 to 5 minute video clips. We have now today with us, Eleanor Brooks is joining us. She produced one of the first ones. What the students have done is they take a full presentation and they summarize it in a video clip. It's a fantastic, fantastic educational tool that can enrich your lectures and your own courses. So please visit the website, email us if you want to be added to the list that gets all the announcements. Of course, the central feature of the Find series has been our speakers, who devote and dedicate a full hour to presenting, and then they join us for discussion, and they have been meeting with us on a regular basis and with our students. For this semester, for this opening session, I chose to see what we had accomplished in Find on the topic of parental care. So here you have the list of 11 speakers who discussed parental care in humans, monkeys, fish, birds, insects, amphibians. So Find has really made it a goal to cover a broad taxonomic range and also methodological different approaches. I call attention on the bottom right to a talk that's coming up on October 29th this semester by Lauren O'Connell on family dynamics in a biparental amphibian. I invited some of them to join us, especially today, and I hope they'll be able to stay for the discussion. I know that Stephen Trumbo and Alex Offit are already here with us. In relationship to parental care, what I want to do today myself is talk a little bit about one particular type of parental care, which is paternal care. And so what I want to do in the next 20 minutes or so is present to you what we've been learning about Titi and owl monkey fathers. And for the first time, I'm trying to take a lifelong perspective on the evolution of paternal care, as illustrated by these 2 taxa that I've been studying for close to 30 years. So, Find takes a comparative perspective. We take also a comparative perspective, sometimes more limited to just focusing on primates, when as biological anthropologists, we try to understand something about human behavior today and the evolution of human behavior. So to this audience, an audience that is so familiar with the evolution of mating and social systems, it is no surprise you are aware that very frequently in all kinds of taxa, whenever we see that male and female establish a stable relationship, develop a pair bond, it is quite common, for example, in birds, that what follows from that is that both partners share in the care of the young. That isn't as common among mammals, and that is somewhat common in some species of primates that happens as well. So one of the questions that has been driving my research all these years is trying to understand the connection between pair bonds in non-human primates with fatherhood and paternal care, trying to see if our studying of non-human primates can shed some light on our understanding of humans. And this started now a long time ago. We had just finished our PhDs at UC Davis with Tony Di Fiore there on the top right. And Tony had already started doing research in the Amazon of Ecuador, looking at some primate species that had multi-male, multi-female systems. And I had started working with the owl monkeys in Argentina. I had done my dissertation with the Titi monkeys. We started talking, and now more than 20 years ago, we decided to develop a project on the comparative sociology of monogamous primates, focusing on the sucky monkeys of the Ecuadorian Amazon, on the Titi monkeys that we were hoping to study both in the wild and the field, and the owl monkeys both of Ecuador and Argentina. So some of what I'm going to be describing today has really resulted, much of the methodological approaches are thanks to this collaboration that we've had with Tony for over 20 years. Most of the data I'll be referring to comes from the Owl Monkey Project of Argentina. And I wanted to get my thanks out there from the beginning. Marcelo Rotundo, the field manager, has been with the project now for 25 years, and he's been an instrumental player in everything we've accomplished. Alba Garcia de la Chica joined us in Argentina 10 years ago, stayed, and today is the co-director of the project. And then the people who are helping us move along these days, Maggie Corley, who also did her PhD and has been doing some fantastic postdoctoral work on genetics. Jonathan Pertil, who's just getting started as a PhD student here at Yale, and David Wood, who spent some time in Argentina, but now is also studying Titi monkeys in Ecuador at UC Davis. Needless to say, all of what I'm going to present to you couldn't have been done without so many students, assistants, and volunteers, and our thanks to them all. What are some of the things that we have accomplished? And I have to say, I'm quite proud that we've done this. From the go, probably because I was trained in the lab, I wanted to have a research program that would really draw from what you can get in the laboratory conditions, controlled laboratory conditions, and in the field. And we've accomplished that to a good extent, both with Titi monkeys and owl monkeys. We have had research done in colonies in the US, where Sally Mendoza, Bill Mason, and now Karen Bales have been doing research with Titi monkeys. We've done research with the owl monkeys that are at the Michele Keeling Center for Comparative Medicine and Research. And of course, then the work in the field with the owl monkeys in Argentina and the Titi monkeys in Ecuador, and more recently in Peru. So I just chose a few references that may help you get an idea of the progress we've made in drawing a richer picture from really tapping on both the lab and the field for the questions that drive our research. The Monkey Project, just to give you a little bit of a sense, those of you who come from primatology, biological anthropology, some of this is, I know it's not new to you, but maybe if you're not into primates, you haven't heard as much about the Monkey Project. We're very fortunate. It's been almost 30 years running strong. We're now studying the 4th generation of identified individuals. We have all kinds of genetic samples available, as listed there. From the beginning, we made it a goal to really have large sample sizes, and we succeeded on that. We do behavioral data from roughly 10 groups. We collect demographic life history data from 25 groups. We have now almost over 300 individuals for which we have very detailed data on their lives. Simultaneously, we collect abiotic data, forest ecology data, and something that Tony and I started early on. We knew that there would be a lot to be gained from doing telemetry. And we have also tried to do some accelerometry to get a little bit more into the physiology and energetics and proximate mechanisms regulating what we're interested in studying. So let's go now to our players in the story. And I want to get done with just one slide about something that it's quite unequivocal. All monkeys and Titis, always and everywhere, have been described as pair-living and pair-bonded. You see a group of owl monkeys on the left, tail twining. You see Titi monkeys at the UC Davis Primate Center on the right, and 2 human beings holding hands. Pair bonded, I'm using loosely in the sense that I am of the opinion that it's very difficult to find robust evidence for a pair bond in the natural conditions of the field, but we have very strong data from the lab speaking of a pair bond, both in owl monkeys and Titis. So the pair bond, we can live with that. Believe me, that there is pair living, there's pair bond. Some jargon to get out of the way. This is the way I'm gonna be, when I'm saying per living, I just mean that for these taxa, you just have one male, one female, who are spending time with each other, who are socializing, who are mating, who are reproducing. Pair bonded, that is a relationship that's relatively stable over time. And then this is not an audience that needs me to explain the difference between sexual monogamy and genetic monogamy. We're gonna come back to that. And then we talk, when we describe the care system, normally we will refer to biparental care when male and female are doing it. For the talk today, I'm referring to paternal care, which of course, it's a given that you also have maternal care going with the care provided by the male in the group. Now, how about, I said that there's several non-human primates that show a pair bond or evidence of a pair. Now, the appearance of paternal care in primates, it's more erratic. And as you can see, don't try to read the names, just focus on the yellow branches and the blue branches. And when you do that, when I graph on here, a graph that Tony put together a long time ago, you see that paternal care in some form or the other shows up in humans, shows up in some of the primates of Southeast Asia, illustrated by a gibbon. Towards the left of the graph, you see some prosimians, some lemurs that show paternal care, and it's in the center where you see the owl monkeys, the Titi monkeys, and the callitrichids, the marmosets and tamarins. But really, except for some forms of paternal care that we may describe in modern societies, and Sarah will definitely be talking about this, it really comes down to the Titis and the owl monkeys, where you see, without any doubts, I'm comfortable saying that I'm not aware of any other mammal where the male is as committed, as involved in paternal care as we see in Titis and owl monkeys. And this happens in the lab. And on the top left, you have an owl monkey male transporting a baby infant at the Dumont Conservancy, where our colleagues Volovich and Sean Evans have been studying them for 40 years. You see it with Titi monkeys at the bottom left, again, a male Titi monkey transporting an infant, and you see it in the wild with the Titi monkeys in Ecuador, with the Titi monkeys in Peru, with the Titi monkeys everywhere they've been studied, and the owl monkeys that we study in Argentina, and now a very solid research program in Colombia led by Andres Link and his students have also documented without any doubts that we have paternal care. So this is happening. The question is what form it takes and how and why. Because it is a little bit of a puzzle, right? I mean, again, I'm not going to go into the details explaining why it is a little bit of a puzzle. Mammalian reproductive biology is very, very different from the situation that you may have in birds, in amphibians, in fish, where fertilization is external. So with pregnancy, with lactation, with internal fertilization, all that brings together both father uncertainty and a very, very differential investment from the beginning. That makes paternal care somewhat of a puzzle in evolutionary terms. Let me walk you, I'm going to take for the remaining of the talk, really a natural history approach to describing what we've learned about owl monkeys. And every so often, I'll back it up with solid data. But for you to understand this natural history, this is just an Asara's owl monkey life cycle. So you have a group of one male, one female. That's the natal group where the infants are born. You have one infant born per year. You don't have twins, like it happens in marmosets and tamarins. Eventually, this individual goes through infancy, the juvenile period, it disperses, it becomes a floater, meaning a solitary individual for some time. We'll go to that in a second. And then it eventually finds an opportunity to reproduce within one of these social groups I have described, right? So that's the cycle. And what I want to do in the next few minutes is reflect and look at this individual going through its life in relationship to how it interacts with its own father, how it's learning about being a father, how it's delivering as a father. First anecdote, this was a funny one. I mean, we had two new assistants, had never been in the field for longer than a few days, and we asked them to stay there put for a weekend for reasons that I don't remember. When they came back on Monday and we asked them anything fun to share from the field, they said, well, not really. I mean, we saw a birth. What? Well, these two students who had not been really spending a lot of time with owl monkeys documented the first observation of a diurnal birth in any wild owl monkeys. And these are not photographs of that birth, but what this photograph shows you, photographs taken by Maggie Corley of a 2-week-old infant, I think it's quite remarkable. On the left, it's hard to make sense of what's going on, but you can see towards the left, an adult with kind of a reddish color, that's the female, and somewhere in the middle, you have the baby who has just finished nursing. This is a 2-week-old infant who can barely move on its own. But I think it's the picture on the right that tells a fascinating story. There you have an infant who is very clumsily moving and seeking the father. He's just finished nursing and now is going to dad. The photo is capturing, this is not just dad going and grabbing the infant. This is an infant who has only been alive for 2 weeks, and the source of nourishment is mom, seeking comfort in dad. Those are things that happen during the newborn and infant stages. During the birth, the father was not really right there at the time that the female was giving birth. It was a 3-year-old subadult, a male, who was very, very close to the female tract, sniffing, smelling, but the male was maybe 20 meters away giving alarm calls. So maybe that was the way in which he was being a dad already. As infants, both Titi and owl monkeys are consistently carried primarily by fathers. These are data from Titi monkeys in captivity at Davis, collected by my friend and colleague Diane Reader a long time ago, but they make the point very, very clearly. You can see how during the first 4 months of life, 16, 17 weeks, consistently, it is the father that carries the infant the most. It is the father who is most in contact with the infant. And this has been like this all the time. No matter who looks at this data or at these individuals, that's the pattern you find. We had a natural experiment in Argentina back in 2011. For the first time, 2 of the 10, 15 groups had twins. So we were very curious, and we just rushed to get some additional funding, which we got to study this process in detail. And let me just show you with the video what was going on. There we go. This is a male. That's the male carrying twins on its back. The 2 twins probably weigh 30% of the male's weight. You cannot see the mother. These twins are maybe a month old each. And it is the male moving them around all the time. The female is going to approach now towards the end of the video clip. That's the female. The female is following dad with the twins on his back. You look at the data, and this is what you get. So what you have here is in purple, the female showing the dotted lines are for how the female in purple and the male in green carried singletons. And you can see that from week 2 to 12, always it is the male who was carrying the singletons more than the female. The solid line, both green and purple, now you see what happened with the twins. It's fascinating because it isn't just that the female did not help more than before. The female even transported the twins less than she would have transported the singletons. So with this natural experiment, all the responsibility, the burden of transporting the twins rested on the male. I should say that neither, in both groups, one of the twins died probably before reaching 6 months of age. And I'll be honest, I don't remember if the other 2 made it. I don't think they did either. These are data very fresh, just from the oven. David was analyzing them in the last week. These are data that come from Titi monkeys in Peru, where David has been studying Titi monkeys for now almost 2 years. This is the same site where some of you may know Sofia Dolotovskaya's research. She also studied Titi monkeys there. And Titi monkey males groom the infants more, as shown by this graph where you have in light blue the frequency or the rate at which the male grooms the infant compared to the female. Again, the robustness of this finding comes from so many different independent studies over different types of Titis, different field sites. The pattern is consistent. What happens when the animals become juveniles or subadults? A very important point for those of you who are familiar with the cooperative breeding marmosets and tamarins. The siblings do not help. When you're a juvenile or subadult, you are not learning how to be a dad by taking care of your siblings. They are almost never involved in doing that. So maybe they're learning through observation, is very likely, but they're not participating. And if you're in a natal group with mom and dad, and you want to become a father yourself, you most likely you'll have to disperse. And this is what this graph shows you. Both females and males leave the natal group. These are data for roughly 50 females and 40 males, showing that both sexes disperse sometime around when they're about 3 years of age. Now, once they disperse, what happens? This is what I was showing you before in the graph. And if you want to watch my presentation in Fine from September 2020, on that one, I focused exclusively on this stage that I'm describing now, dispersing floaters. We've been talking about a pair living group, a female, a male, with a pair bond and producing offspring. Now you leave that group and you become a solitary floater, male or female. And if you're going to become a dad, one way of doing that will have to be to find a partner. And way of finding a partner, what we have learned is that what you do is you try to replace, kick out the same-sex adult in one of these pair living groups and move in. Both males and females are doing this. They're moving in and replacing resting adults to become the reproductive adults themselves. These are data from 10 years ago, just showing you that over the course of 10 years, we saw 27 female replacements. We saw 23 male replacements. So that's something we have not yet documented in other owl monkey or Titi monkey population. So it's still unknown whether our population is truly unusual or it just hasn't happened that we've been able to detect it at other sites. Now, what's interesting is that because of these replacements, when we look at the numbers in our population, about a third of those juveniles and subadults that I was describing to you really grow up in a group where it isn't just a biological family. They grow up with a stepfather, they grow up with a stepmother. And so if that happens, what it may happen to you if suddenly, if you're a male subadult and your mother gets replaced, and now you have a stepmother, you may find that that stepmother becomes a potential mate. So here I'm showing you a graph showing how subadults tend to disperse later when a potential mate replaces their opposite sex parent. Let me just give you quickly another anecdote, fascinating story that's big. I mean, so much richness in this natural history. We had a subadult male who was living with mom and dad, Enrique, who left the group. I mean, he became a floater. And this becoming a floater, it's not a definite stage. Sometimes they go, sometimes they come back. And eventually, during the 3-4 months that Enrique was trying to find his way and trying to find a partner and maybe trying to replace some other adult, his mother was replaced. So eventually he goes back home and he finds that dad is not any longer with mom, but dad is sharing that group with the stepmother. And Enrique finds dad's new girlfriend kind of attractive and has no better idea that starts socializing and shows an interest in that stepmother. So he's coming back. It didn't end up well. There was a lot of aggression now between the father and the son. And eventually Enrique was really kicked out of the group. But so there was a mechanism of inbreeding avoidance. He left. When mom is replaced by a stepmother, he goes back and finds interest in a potential mate. Again, he didn't succeed becoming a father. Once they do become residents, these are data showing you how long they can hold tenure, reproductive tenure, and showing you in blue the males, in red the females, roughly, again, about 50 adult males and females, and you see that their tenure length is rather similar. The males are averaging 5.7, I call them years in the text. The graph talks about mating seasons. The females are a little bit shorter, and there's a lot to be said about that. We get the impression that the turnover of females is faster than the males. But it is during this tenure length that the males and females are going to produce young, that the males are going to produce the babies and that they will take care of. Now, the central question to all this, I mean, I was alluding to the need to understand the genetic relatedness between the partners when I was describing the situation with Enrique. It is even more important to understand the genetic relatedness between the father and the infant in trying to understanding why and how paternal care is provided. It really became necessary fundamental to assess the extent to which what we're describing as sexual monogamy really also meant genetic monogamy. We first published this in 2014 when Hrdy, a colleague, did the analysis of roughly 35 dyads. We looked at 35 infant and puberty fathers to assess if really those adult males were caring for their young or not. I mean, I don't need to tell you that with all we've learned from bird literature and amphibians, all kinds of taxa, extra-pair copulation was very much in our minds, but it didn't happen. For that first analysis in 2014, we could find no evidence of extra-pair paternity. So only in the last couple of years, we have extended that analysis. We went back and looked at samples that we had collected in the last 20 years. We had almost 400 unique individuals. And we did pedigree analysis of 126 infants using 22 microsatellites. And it was fascinating. I mean, I have to say, it's still I find it hard to believe. In red, you have now the new numbers. In black, from the paper published by Hrdy in 2014, the original sample size, I mean, it was 17 pairs that we looked at. Now we have looked at 59 pairs, 126 offspring, and not once were we able to rule out the male in the group at the time of conception as the most likely sire of that young. So genetic monogamy, the males taking care of young that they have unequivocally sired. And then, of course, you start closing the loop and seeing how this strategy of providing a lot of care to young may be adaptive. It shows in some data on infant survival, which are very, very high. For the 186 infants on which we collected data, you have roughly 87% of infant survival. And for the data on Titi monkeys, these are data from Titi monkeys in Ecuador that we collected with Tony. 22 of 24 infants survived. It is my understanding that the pattern holds for the Titi monkeys in Peru, where Sofia Dolotovskaya, I think she has data close on 10 or 15 infants. And David, in the last 2 years, he was telling me that 8 of the 10 offspring that were born during his study also survived. Oh, here it is. I got ahead of myself. A final result that I wanted to share with you is another angle to looking at the benefits of having your biological father or mother take care of you. But the main point here is how important fathers are. On the top panel, we see the survival probability in the presence of a biological father or stepfather. And you can see that the drop in the survival probability is bigger when your biological father is replaced, top panel, than when your biological mother is replaced. So there are more negative consequences for an infant of losing the father than losing the mother. We just don't know what it is. We know the outcome, but we have not really been able to put our fingers on what is it that the infant who loses a biological father is missing that affects its survival into the future. So to wrap it up, I'm doing well with time. To wrap it up, I'm going back to the title. This is where I'm going to start transitioning to the future and to Sarah's discussion and the importance of all this, of the comparative study of paternal care in non-human primates to understand something about human dads. The title says, On the Evolution of Paternal Care. And every day I feel more and more uneasy about having the word evolution there, because I see that even after such a long time and so much data, it isn't very clear what we have actually learned in a very, very hard sense about evolution, the way we discuss evolution in our introductory courses. Yes, proximate causation, proximate mechanisms, but I want to share something with you, hoping that it will give us something to discuss in the second hour. These are some chapters that discuss the relationship between owl monkeys and Titis. So I've been talking all these 20 minutes about owl monkeys and Titis. And the behavioral convergence, the behavioral similarities are absolutely astonishing. So in the book that I edited on owl monkeys last year, there's a chapter where Alfie Rosenberg and Marcelo Tejedor focused on morphology, and they are of the opinion that owl monkeys and Titi monkeys are closely related based on their morphology. For example, Alfie would marvel at the photos that I have of both taxa tail twining. And that's their position, that's their opinion based on morphology. But every single piece of genetic data that my friend Tony Di Fiore with his students Lynn and Amelie reviewed in amazing, these two are two amazing chapters. You may agree or disagree with their conclusions. Every single data point on genetics suggests that owl monkeys and Titis are not closely related. Quite the contrary, they're rather apart for some substantial evolutionary times. So as a behaviorist, I'm in the middle and I'm just puzzled. I mean, really, I'm inviting you, please email me. It'd be fascinating, in my view, to have over the next 10 years, somebody who doesn't have the baggage of being a primatologist, look at this relationship, look at these similarities between owl monkeys and Titi monkeys in so many aspects, and see the extent to which we can understand better what are the proximate mechanisms that are making them behave in this way if they are not closely related. Is this a case of convergence? So this is an invitation to continue the discussion later on. I'm sorry, because they are pair living, they're pair bonded, similar size, territorial, and fantastic dads. So what makes them fantastic dads? We don't know what makes them. We know they are. We know that it's paying off in terms of adaptive traits. Things take a long time. I just wanted to close with this one. It's fascinating. I was reading, Sarah makes reference to this in one of the early chapters of the book. It's been 45 years that Sue Carter and Lowell Getz got together and published their first paper in 1981 in Behavioral Ecology and Sociobiology. Look at that first sentence in the summary. They were just providing some evidence for the existence of pair bonding or monogamy in prairie voles. Now, 45 years later, we have made huge progress in understanding not just pair bonding in voles, but the role of oxytocin in regulating social relationships. So yes, these things take a long time. I'm hoping that our data on natural history, our solid data describing the patterns that we see will help us make progress in understanding how it happens that Titi monkeys and owl monkeys are such amazing dads. Very quickly, you don't need to see this, but of course, thanks our funding agencies. And with that, I want to stop my presentation and move us to really what I think is our treat for today. I want to introduce Sarah Blaffer Hrdy, my colleague, my friend, and one of my mentors. We could spend a whole hour covering Sarah's history, but I know that we're going to benefit even more if instead of me talking about her, we let her talk. Let me just say that she considers herself an anthropologist. She's been working on the evolution of primate behavior for 40, 50 years. She's published 7 books. When you look at her CV, I thought, well, what can I say that we don't get to see frequently? Well, she's got 4 honorary PhD degrees from very prestigious universities. I think that is something that I've not heard before, or at least not that frequently among fine speakers. These are some of the books that you may or may not have seen, but the ones that are most relevant to our meeting today is Sarah's trilogy on parental care. She first published Mother Nature in 1999, Mothers and Others in 2009, and now in 2024, Father Time. It takes 10 years to produce these high quality volumes. It takes 10 years or 15 in the case of Father Time to do the thorough research that Sarah has done to inform, to synthesize everything she's learned about Father Time as a natural history of men and babies. So with that, I'm going to stop sharing and I'm going to invite Sarah. Now I'm trying to recenter and find Sarah on my screen. Where are you, Sarah? You're there. So with that, I want to invite Sarah to take it from here. And in case you need to go, remember after Sarah, we're going to move on to doing the raffle. We hope to be done by noon with the raffle. And after that, I hope you'll be able to stay to exchange in an interesting discussion among all of us. Sarah, I cannot thank you enough for having agreed to join us. And please, it's all yours.
Thank you, Eduardo. That was a wonderful talk. And of course, thanks to Karsten, Lauren, Adriana for these wonderful fine talks, which have been so important to me. And thank you for the opportunity to comment on the relevance of the kind of research you're doing for understanding the human condition, which isn't easy. My single biggest takeaway from your talk was how from 28 years of hard-earned behavioral and genetic data, you've learned that the males who remain in a monogamous pair bond with the same female have a 25% higher reproductive success than a male with successive mates, which is just extraordinary. And what it shows is that genetic monogamy, under some circumstances, pays off. Had that been the case for our hominin ancestors, humans would be a very different species from what we are. But prioritizing direct care of babies the way Aotus azeri males do is rare. Instead, the mammalian default is, of course, maternal care, typically exclusive maternal care. And why not? I mean, for 200 million years, she's been there, present when the baby appears. She gestated that baby, was hormonally primed to care for it after birth, and she provides this extraordinary substance, this white gold, tailored to her baby's nutritional and biogenomic needs. Of course, we have maternal care in mammals. That said, the prosimians, monkeys, and apes, and the highly social order primates do tend to have more allomaternal care than is found among primates generally. Carnivores do too, but it's especially prevalent in the social primates, where about 40% of species of primates have some degree of allomaternal care, and males are doing quite a bit of this. The thing is, this isn't really direct care of babies. It's typically a male who's present in the group, rescuing an infant in trouble, or perhaps adopting an older orphaned infant already near weaning. Otherwise, the male care of infants, little babies, really doesn't amount to much. And people who wonder why I focus so much on babies rather than father-offspring relations in general, that's simply because the animal behavior data on males and babies is so much better. We don't really know what the relations are like for most animals with older offspring. The exception you mentioned, of course, was siamang, a lesser ape. We haven't had an ancestor in common with them for 17 million years. And there, after at least a year, the mother turns her increasingly heavy, somewhat increasingly independent infant over the male to carry most of the time. This is Susan Lappin's work. And as a consequence, the female is able to restore her energy and breed again sooner. So the mate benefits. And siamang are mostly monogamous, but there is polyandrous mating under some conditions. They're not quite aotus quality monogamous. Otherwise, statistically speaking, the closest a chimp, orangutan, or a gorilla male ever gets to a brand new baby is when they are mauling or cannibalizing a baby. You do not see direct care, that just urge, I've got to get my hands on this baby, the way you see in Titi and owl monkeys. And that's apes, other non-human apes. But when you get to humans, all bets are off. Different men in different countries, different cultures may care a little, a lot, or not at all. Even some men certain of paternity act as if they didn't even know they had children, and some of them don't. This is why along comes the 21st century, and I'm just astounded to see these men, some right in my own family, devoting their lives to these babies, taking over half or more than half-time care of babies, taking advantage of new technologies, rubber nipples, baby bottles, safe formulas, breast pumps, to care for babies right from birth, sometimes with no woman involved at all. Well, I grew up in South Texas, Eduardo. And, 8 years ago, I had never even seen a man back then changing a diaper. And now this is happening all over. That boot camp you've set up in Argentina, all these men caring for babies. It was astounding. And it's true that as early, back in 1980, Alan Dixon reported higher prolactin levels in males caring for babies in marmosets. And people didn't believe him at first, but, it was replicated. And by the end of the century, Karsten and others were talking about prolactin as the hormone of paternal care. And then following ornithologist John Wingfield, people often started to expect testosterone to go down after males switched from the challenges of the breeding system to having to tend their mate's offspring or helping to. But very little of this research focused on men. It was like we just didn't ask the questions. And it really wasn't until two animal behaviorists, Anne Story and Catherine Wynne Edwards, up in Canada, couldn't get the funding they needed to study paternal care in their various rodent species they wanted to work with. And they had to study the fathers at lower cost, the fathers at lower class in a local Lamaze class, and reported that, sure enough, prolactin levels go up, testosterone goes down, surges in oxytocin, and then it was like the floodgates opened. All over the world, animal behaviorists, neuroscientists, biopsychologists were paying much more attention to what was going on in fathers. Alison Fleming in Canada, another animal behaviorist, was also beginning to look at it then. But this was really the start. And from then on, there was this rush to study paternal care in humans. I need to say, though, that even before the 21st century, anthropologists like me were aware of how facultative male care was in humans. And we also had the advantage of the wonderful research by human behavioral ecologists working with African hunter-gatherers to get some idea of what challenges hunter-gatherers on an African savanna encounter keeping their babies alive. And it was clear that males were helping, but that a single male all by himself, man the hunter provider, couldn't meet the terms of that so-called sex contract. You give me what I need to raise my baby, I'll give you certainty of paternity. The men just couldn't meet the terms of that. And their findings convinced me that there was no way, and this is when I was writing Mother Nature, but then followed almost as an appendix to it with Mothers and Others, there was no way an ape with the life history attributes of Homo sapiens, that is, our really costly babies taking so long to mature and having to give birth after shorter intervals because they faced what Lovejoy back then called the demographic dilemma of, and all great apes have that, by breeding so slowly and as their environment gets tough, they start to go extinct. Well, the other bipedal apes out there all went extinct. And the slow reproduction was surely part of the challenge they faced. And so that's when I became convinced that alloparents, that is, group members other than the genetic parents, had to have helped care for and provision babies in addition to parents, even if both parents were present. And this is, of course, what sociobiologists back then called cooperative breeding. And across the natural world, cooperative breeding has evolved in, maybe 10% of birds, but it's only found in 5% of mammals. And even though the incidence of shared care is much higher in social primates than it is in most mammals, shared care accompanied by extensive alloparental provisioning is only found in some of the marmosets, some of the Callitrichidae, tamarines, and in humans. Even in Euryiotis, am I right, Eduardo? It's kind of limited provisioning. It's just not much. Yeah. And this, I suspect, though, in humans was just a game changer. Sharing food became absolutely critical for survival, and it enforced a kind of mutual dependence on people that changed, I think, the terms on which really powerful evolutionary forces like sexual selection were acting. They still go on, but social selection becomes more important. People are interested in what other people think about them and their reputations and so forth. But that is another very long story. But if correct, this hypothesis means that shared care and provisioning of offspring among members of the genus Homo was co-evolving right at the same time that brains were getting bigger and our distinctively human frontal cortexes, especially the things like the medial prefrontal cortex, were evolving. And this is why in 2014, I was so riveted when I read Ruth Feldman's team's findings from one of the first neuroscience studies to scan the brains of fathers helping mothers, showing that the parts of the brain that were most activated were in the frontal cortex. And this is what I was already suspecting, that humans had this alloparental substrate in their brain to make them very susceptible to cues of need from infants. And this was completely consistent with that. But the really galvanizing thing was their companion study, where they looked at what was happening in gay fathers, where two men were raising a baby by themselves, with no mother involved at all, no woman involved at all. And only one of them was definitely the father. And they were, of course, using modern technologies to do this, formula, baby bottles. But in their brains, not only the newer portions of their brain were activated, but also the most ancient portions of their brain in the limbic system. We're talking about the amygdalas and hypothalamus, the same areas of the brain that are activated in mothers. Well, how could this be? And this is when I set out to write Father Time and chapter by chapter hypothesize what must have happened over the course of mammalian, primate, hominin, and human evolution, and in modern human history to make this possible by the 21st century. And of course, when I got to the apes in the line leading to Homo sapiens, one of my big challenges was to explain how a mother could ever trust anybody else in her group, male or female, enough to turn her baby over to that individual, to allow them close to her baby. Because males in most apes are an existential threat to new babies, not a source of help. Well, this is where the work on hunter-gatherer subsistence modes and their unusually flexible resonance patterns come in. They're even more flexible than your aotis in terms of males and females moving around. By the end of the Pleistocene, though, with the adoption of novel subsistence modes, very different family structures, customs, and ideologies were arising. And over time, they were changing, and over further time, changing faster and faster. I mean, just in my lifetime, so much has changed. And this is where the work of social historians, economists, legal scholars proved essential for fleshing and morphing out legal systems, new educational and economic opportunities for women. We can own our own property in Western societies now, something very new, and earn good livings. We can support young on our own. And morphing legal systems so that we can actually have title to babies. All this is new, and it was relevant to what was going on. But I still needed to explain what those molecules like oxytocin and prolactin and galanin and dopamine, along with the relevant neural circuits for getting involved in babies and turning on your ancient neural circuits for nurturing, what were they doing there in the first place? And here I was guided by evolutionary theorists like Mary Jane West Eberhard, who Eva Fischer talked about recently. And here I was also guided particularly by her ideas about the importance of phenotypic flexibility and her insightful speculations on cross-sex transfers and how this can happen. I could rely on the highly innovative research on antagonistic neural circuits generated by Michael Newman and Kumi Kuroda in Japan. I think she talked in one of the fine talks, but not about this. Catherine Dulac at Harvard and other neuroscientists, along with the absolutely superb work on flexible parenting in poison dart frogs that Eva Fischer, Eva Ringler, and on October 29th on poison dart frogs that we're going to hear about from Lauren O'Connell. And it's early days for understanding what's going on here, but we need animal behavior to figure out the human condition, it turns out. The existence of antagonistic neural circuits mediating male infanticide, feeding, cannibalizing babies on the one hand, and nurturing them on the other. Understanding this would have been just impossible. The idea of it would have been inconceivable without first knowing about the challenges faced by early vertebrates, among whom male care first, in fact, parental care in general, but male care in particular, first evolved over 400 million years ago. This is not something I was thinking about 25 years ago, but I am now. One of my goals in writing Father Time then was convincing fellow social scientists of the importance of integrative sociobiological approaches that draw on natural history and comparative perspectives to inform our understanding of the social and cultural contexts in which neural circuits relevant for parental care are activated. Along with cultural histories, these much deeper natural histories are profoundly relevant for understanding parental decision-making and infant and child development today. Those in a wide range of disciplines stand to gain from seminars like the ones it finds, just as this anthropologist clearly has. So thanks, Fine.
Thank you, Sarah, so much. Lots and lots of thoughts, I'm sure, in the audience that we will be able to listen to as we move on to our second hour. But now it's time to do another little bit of celebration. We in Fine, and we're going to move on to doing the raffle that we announced. We ask so that we feel you are with us, that if you're going to participate in the raffle, we invite you to open up your video and make sure you're not a bot, and bots don't get books. So we invite you to show your face and be part of the community. I want to thank David and Alba. Alba wasn't feeling very well, so she may have left, or she's there behind the scenes, knowing that we can count on her if need be. They were responsible for working out the logistics, make sure the raffle goes well. So David, I invite you. What I'm going to do now is I'm going to open, the way it's going to go is I'm opening a poll right now. And the poll is going to ask that you enter your full name in item number 1, and then you're going to enter your email in item number 2. Everyone is invited to enter the raffle. Don't be shy. If you feel you're senior, etc. Let's have all fun. Once I take all the questions in, I'm going to close the poll. I'm going to end the poll, and I'm going to share a file that I can easily download with your data. You give it to David, and David is going to spin the wheel. We're going to spin the wheel 7 times, 1 for each book. The books are going to be, David knows the order. You can only win 1 book. So if you get a second one, we'll skip you and we'll spin again. So I'm going to give this a couple minutes. The answers are coming in. I have 23 or 50% of you participating. Still coming in. Some of you have left. Right, we have 50 participants in Zoom, and it's counting 44. I don't know why it says only 44. Still coming in. Let's give it a couple minutes. I want to be sure everyone's name is in if you want it to be in.
I want the Owl Monkey book.
Well, we'll get you one. But no, go ahead, enter the raffle as well. I mean, let's still have some fun. You can relax and be.
I know. That's why
I've heard from some of you who told me, no, I'm not going to enter. Come on, all of you. I cannot. I was trying to type, and I guess because I set up the poll, I'm not allowed to enter my name. But I really mean it when I say, all of you, come on, enter your names.
Eduardo, how is Fine financed?
With our volunteering time.
We shouldn't be paying to be part of the raffle.
No. This is, I mean, if any of you have ideas on how we can find some little support for the let's agree that not everything is fun, right? There's a lot of behind the scenes that are not fun. But they become fun when we manage to put this together. So to I have 38 responses, 48 of you in the audience. I'll have a quick look at your faces. Anybody give me, if you feel like you have an answer yet, I think we're ready, David, because I got 38 responses, and you've had 3 minutes. If you couldn't type your name in 3 minutes, you're not going to be able to read the book. So, so I'm gonna I'm gonna end the poll now, David. Do you agree?
Yeah, sounds good.
We're ending the poll. I'm gonna download the results. There we go. We copy quickly. I go to the Zoom, and we practice this more times than you would imagine. So I need to attach the file. Oh, I was about. Give me a second, please. David, there you go. Okay, David, I just pasted the file in the chatting window just in case. Alva, I'm sending a message in case something goes wrong to the two of you with the file as well. But David, you should have the file already.
I do.
What I can tell you in the meantime, while David sets up, what's going to happen is that he's opening this, he's reading the CSV file into the software they found. And so the wheel is going to have the 38 names of those of you who entered the poll, and he's going to spin the wheel. We're announcing, okay, now we're going for book X and Y, spinning the wheel, and we'll see what names comes up. And after that, we'll spin the wheel again. So, David, do you want to share your screen?
Yeah. All right. So this
There
first
you go.
book.
That's transparent.
I know.
This first book will be Mammal Societies.
Mammal Societies contributed by our past speaker, Tim Clattenbrock.
Here we go.
And I will be announcing the winner in the chatting. Alex, right?
Mm-hmm.
Okay.
Hooray!
Congratulations, Alex.
The next book is The Evolution of Primate Societies.
Peter couldn't be here. He's somewhere else. Martha.
The next one is The Evolution of Social Behavior.
By Michael Taborski. Jane.
And next is Slow Birding.
Slow Birding, the new book by John. Tomislav.
And now Die Biologie des Vaters.
Okay, bear with me.
By Karsten.
I have no idea. Something like this, maybe? Sayantandas. Sayantandas.
Next is Owl Monkeys by Eduardo.
Nata.
And now finally, for the grand finale, we have Father Time by Blaffer Hrdy.
Hrdy.
All right. That's it.
Okay, so I need to remember to say the chat or somebody.
I have the list.
You have the list of the winners.
Uh-huh.
Okay.
Are
Well,
you going to send us the addresses?
we will
Yeah.
email you if you want one of the books. Somebody will be in
No,
touch
no.
with you.
I need Hrdy Goodwin's address.
Yeah, we will reach out to all of the
Well-
winners to get mailing addresses and deal with all that after the meeting.
Right. Well, thank you all for entering the poll. Thanks for joining us for the presentation. And we can move now to our second hour, which I really think, I mean, all of us, Carson, Adriana, Lauren, the students feel like the 1 hour discussions again and again, we keep hearing that it's really been a treat. It's really been a signature of the series. And we're not going to change from what we've done in the past, which has been explicit about our invitation to our young scholars to jump in and ask questions and share their thoughts. And the best thing that we can do today as we start a new season is go with that. So as usual, you can type a question mark in the chatting window, and I'll be taking the questions as they come up. But if you're a student, type an S by the question mark, and we'll most definitely give you priority. So we're opening the floor to questions or comments. We have some, Sarah's here. I'm trying to think. Steve is here, Steve Trumbo, who discussed parental care in burning beetles in the past. I don't know whether Alex had to leave, but can be a question to Sarah, to me, to anyone in the audience, can be a reflection of yours. I don't see any questions in the chatting. Come on, guys, I ask. Rag, Rag, you need to be second. I want to hear from a young voice first. We're going to be waiting. Come on, guys, don't be shy. I'm tempted to do what some of you have told I've done in class. I just leave the classroom and ask the students to call me when they are talking, and it works. I could leave the session, but it may not work. No one? Well, Rag, you go.
Okay. Thank you. I enjoyed Eduardo's talk and Sarah's comments. I've read her book and I've read your paper, so it was a wonderful way to reflect back on all of these things. One question occurred to me, Eduardo, when you said that there is a more drastic mortality when the baby loses the father than when the baby loses the mother, right? You gave some data that the mortality is even higher when the baby loses the father than the baby loses the mother. And I saw that there was one confounding factor there, because whenever a parent is lost, there is a step parent present. So I was wondering whether it is the loss of the father or the presence of the stepfather that's causing this high mortality.
So, well, I think we're doing well if you only picked up on one confounding factor.
Mhm.
So, since there are probably a gazillion of
Sure,
them,
sure,
and
sure.
we know, but this is the time where we can go a little bit into more detail. And I'm not gonna, I know Alba, like I said, she's not feeling well. Otherwise, I'm sure she would be delighted to chip in. Please, Alba, if you feel okay, jump in, because you know this analysis better than I do. First, we talk about survival and mortality. None of these measures are so unequivocal that we actually, what we find with the analysis of the data, and my goodness, have we looked at this data, is that definitely there is survival rates higher. Survival rates are lower when the biological father is absent.
Yes.
But you're right. Something that I didn't say in the talk is that it never happens that a group ends up with only one parent in the group.
Mhm.
I talked about the replacements, and those are kind of forced evictions, but it also can happen that a male or a female dies of natural causes. And it's remarkable. Ours is a very saturated habitat.
Mhm.
I think I can count with the fingers of my hands the number of hours or days that we've seen only one parent. Somebody
Mhm.
moves in right away. So
Sure.
anytime a biological father or mother is gone, a stepmother or father is in. Now,
But
we
I'm
have
wondering whether
no,
you have any behavioral data.
right, we have little behavioral data on what happens with those step parents. Nothing like Sarah was describing for some of the apes. No clear evidence at all of
Mhm.
negative interactions between the stepfather and the young. No
Mhm.
clear negative. I mean, we have some little data on agonism, very broadly defined, and very
Mhm.
small cases where apparently the step parent, mother or father, may be more agonistic than the other one. The way I've started to think about this, I think it takes me back to the lab where we can have, and this is where we're really hoping that David Wood's dissertation looking at the interactions in the lab will give us the nuance, the detail.
Mhm.
So what I'm making of this is that it could be that it's the loss, it's not the presence of a stepfather, but it's more like what are they missing from the biological father? And that
Mhm.
we still don't know, because remember that the truth is that the effect of the loss of the biological father is more manifested when they are juveniles, when
Mhm.
they're not as dependent than
Right.
when they are dependent.
Mhm.
So what is it that the infant or the juvenile is missing when the father is gone? We just don't know. Now, you go to the lab, and something that I didn't say, but the photograph that I showed speaks to it. From all of my colleagues who have kept colonies of owl monkeys and titis, what they will describe, and this is in writing, I'm thinking the work of Karen Bales, I'm thinking the work of Sean Evans, is
Mhm.
that it all comes down to how tolerant males and females are of their young. And
Mhm.
it isn't just that a that the mother doesn't seek the infant or the father. There's a big difference between mothers and fathers in how they seem to be interested in the infant per se. I mean,
Mhm.
we hear from people taking care of colonies of wild monkeys and titis that again and again, the mothers are somewhat agonistic and aggressive towards their infants, who
Mhm.
find in the male a source of comfort that they may not have with the mother after they nurse.
Mhm.
That's a long answer. We have no clear evidence of the step parent, male or female,
Mhm.
being actively aggressive or
Mhm.
actively negative to the
Mhm.
infant.
Thank you.
Now I see questions, Mark. Okay, Karsten.
Yeah, you could first, hello, you could first ask Kaya, as you said you want to get in.
Kaya. Thank you, Karsten. Kaya.
I was hoping for a little more time to think about how to word this, but well, thank you for both of your talks. I have kind of a vague question, and I guess both of you alluded to, why have a high level of paternal care, facultative or otherwise, in these certain mammalian species and not others. And why questions are always really difficult in evolution, but and potentially impossible to answer. But I gather that I guess some kind of K selection element or the sort of long life history is a part of it. And then one might ask sort of why do other mammals that have, few offspring that take a long time to mature, why would they not have paternal care? I'm thinking about bats, and maybe I'm wrong. Maybe there is paternal care in bats as well, or allo mothering. I mean, I've heard of some instances, but have either of you thought about the sort of why not aspect when you're thinking about hypotheses, and what have you gathered from those musings over time?
I'll answer very briefly, and then I'm going to invite Tony or Sari, both of whom have also been thinking a lot about Sakis. When Tony and I started drawing this comparative study where we would study Titi monkeys and all monkeys. We knew that they had a very, very extreme form of paternal care. We also knew that the Sakis, which are found in the same habitats as the Titi monkeys in all of the Amazon basin, they are almost 2, if not 3 times larger than the Titi monkeys. They have more sexual dimorphism, but there's no doubt. I mean, we're not finding 100% agreement on the relationship between the Titis and the owl monkeys, but definitely we have 100% agreement on the phylogenetic relationship between the Titis and the Sakis. And the Sakis, so we said, wow, this is so cool, right? Because we can look at the same ecology. We have the Titis, we have the owl monkeys, we have the Sakis. And the Sakis do not show anything, nothing compared to what you see in the Titis and the owl monkeys. So I hear you, and I think that's, my hope is that in the next 20 years, we'll be able to understand some of the genetics underlying any of these. I mean, I'm hoping that finally we'll make in the next 20 years some progress in the behavioral genetics that applies to primates. But I really want to hear from Tony and or Sari. What are your thoughts on how you would answer Kaya's question about absence of for the Sakis? And of course, definitely, if there's someone in the audience who can tackle the question about bats or other mammals, please, we want to hear from you.
I'd love to address Kaya's question because it's a good one.
Go.
Can you hear me? Okay, I wasn't sure it wasn't muted. I mean, you're getting at sort of approximate and a situational level, both there. And in terms of the sort of ecological conditions and the situation, there has to be something that a male can do that contributes to infant well-being. And of course, in humans, care and provisioning, it's obvious that that is really helping. But the other thing that I think you really need, and this is at approximate level, and it's what in Father Time I talk about as time in intimate proximity. And it's this tipping point that in humans seems to be correlated with these particular endocrinological and neural effects of, prolactin going up, oxytocin surging. This is time and intimate proximity that is doing this. So the question I had for humans in the 21st century was, what is it that has changed about our residence and subsistence patterns and family structures and ideologies and moral systems that promotes men to be spending more time in intimate proximity to babies? And I even have an acronym for it, TIP, time and intimate proximity. It's like this tipping point. And it seems to be the case in, when you look at Sue Carter's, you mentioned Sue Carter's prairie voles. Those prairie voles, for various ecological reasons, are remaining with their mates. Over time, this put them in close proximity to babies. And in the case of prairie voles, there apparently was enough evolutionary time, enough generations there for selection to act on the males who were able to contribute to infant survival in a way that affected male reproductive success. That hasn't happened yet in humans, and it may never happen. This is what I'm writing about in Father Time is what's possible to happen in males, not what's likely, unfortunately. Does that help at all?
That was a great answer. Yeah.
Anybody else
Especially
who wants
you need
to-
something for the fathers to be able to do. That would answer the question probably, with the bats and so
And
on.
the
with
same,
the
the same goes for grandmothers. You've probably heard a lot about what human grandmothers are doing in humans. That's true. But, in other primates as well, like baboons and chimps, and certainly the langur monkeys I studied, females have so much donative intent. They're decrepit. They're just outcasts in their group almost, and just not doing anything. And all of a sudden there's a threat, like an infanticidal male or an intrusion from outside. Those old females are doing it. But they can't do that all the time. But in humans, and this is, of course, based on the work of Kristen Hawkes and Nick Blurton-Jones and James O'Connell, there's something they can do every day because those old females are bringing back in food. And that's why food sharing is just such a game changer in humans. And there's something males can do every day, which is unusual.
Great answer. Thank you.
Tony, how long ago? What's the divergence time for Sakis and Titis?
Probably 18 million years, something like that.
And what's the divergence time? Alex, I saw you there. I mean, do you know the divergence time between the various kinds of voles, those that the pair-bonded ones are not, and the not pair-bonded ones? Anyone knows how long ago they diverged?
Oh man, you're putting me on the spot. They certainly a handful of million years, but I don't think it's quite as deep as what Tony just said. Between, say, the really the prairie voles breaking off from the others, because it's really just the prairie voles that show a form of monogamy and parental care.
But given how facultative some of these traits are, this is where the work of people like Mary Jane West Eberhard becomes so relevant. I'm not sure that it takes quite as long as we're thinking. And I'm reminded here of those wonderful, wonderful early studies Bill Rice and guys did with Drosophila. And of course, Drosophila, when they're mating, are delivering these toxic molecules in their semen. And these make the females breed faster, yes, but it also makes them die sooner. It's not a great sexual favor. And when he did this extraordinary experiment, forcing some of his Drosophila to be monogamous for just 44 generations, the males quit producing the toxic substances. I mean, that's just 44 generations. Admittedly, they're flies, but like in the poison dart frogs, their genetic setup is such that they too can evolve this stuff faster, and it's already facultative. Because even in the prairie voles, someplace males are carrying more than in other places. I think up in Newfoundland, and Story had kind of a different story for her voles. So I think we're still finding out how long it takes, and I have put my neck out there suggesting that 2 to 3 million years was enough for cooperative breeding to really make social selection more important in humans than it is in other apes. But I may be wrong.
Sarah, when your original comment a moment ago had me thinking exactly the same, along the same lines, where I was thinking within our prairie voles, we find not just geographically, but within our same population, profound amounts of variation, both in terms of propensity to form bonds, but also degrees of parental care that we see. And so you can imagine, I really think that you're exactly right about the fact that within just a handful of generations, you could get that to evolve if you have the right sort of substance to work upon, sort of latently in the brain. And of course, we know there's some big differences between the mammalian maternal brain and paternal brain. And yet there are also there's a Venn diagram there of shared mechanism of which that can evolve out of. And so, yeah, and in fact, yeah. And then, your explanation for why it might arise in humans was a question that I wanted to ask. So I'm really appreciative of Kaya for asking that question, because, and your answer reminded me of sort of a paper that Jerry Wolf and McDonald wrote, saying that they're mate guarding, and they're stuck there at the nest. So why not start to take care of the offspring? I mean, that's one of the many pathways in which you could imagine the evolution of paternal care. But
that's the pathway I see as well. Yeah, absolutely. Yeah.
Karsten.
Yes, first would like to comment to the previous question, even though my comment relates to what I want to ask Sarah. And when we look at at the phylogeny, Eduardo, you showed of the primates, it seems quite obvious in primates. It's some separate branches of the trees. There are some pair living species that might even have paternal care. And we then believe this is all something very special that has evolved there. But when we look at mammals in total, I think that paternal care might be much more common than people believe, because most studies are on large mammals. But when we look at small mammals, it might be much more common than people so far believed. There has been some old studies showing a lot of paternal care in rodents in captivity. Then they claimed it cannot really be, these stupid rodents cannot be paternal. It must be an artifact, because we have this cultural idea that pair living and paternal care must be evolutionary so advanced. And we, that's Laura and myself and co-workers, have shown that in many mammals, pair living might be much more ancestral than we believe, and the same could be true for paternal care. And I think there might be quite a big influence of our cultural background, how we look at all these patterns and what we expect. And that's what I would like to ask Sarah. I mean, she talked about now it's so new that human fathers are so attracted to their infants and so such attachment. But if we don't look at this pattern, but at what we in evolutionary biology call parental or paternal investment, is this not much more common in all human societies? When we look at warriors of the Maasai or the Zulus, they might not traditionally go and hold the babies, but they might still invest a lot in resources for their infants, for their offspring, for the juveniles, the teenage kids. For Zulu, I know it's very important, the status and the world of his father. And if you look at investment, I think in many human societies, or societies where fathers might not be the ones who change the diapers, they might invest a lot. And this is also kind of paternal care. So that's my question. When we go further, including paternal care or the investment, might it not be much more common in human society than just a few societies where fathers started to change the diapers?
Do you want me to answer?
Yes, I didn't read your book yet, so but I guess maybe you have something in your book that human fathers are not only characterized by changing diapers, but by direct investment.
If you read the book, you'll know, first of all, I agree with you profoundly that we mainly see what we expect to see, and that this shapes our science, and that we absolutely have to be aware of our biases, as you and Eduardo are constantly reminding us, take your limitations into account. And it's a very personal book, because I'm laying out my biases from growing up in South Texas and then a Harvard education, where there was a very fixed notion of what males were doing and what females were doing. And you ask why about older ages. I think you're absolutely right. Men are doing different forms of investment all through the ages of their children and through their own life courses. But in Father Time, I made a conscious decision at the beginning that I was going to focus on men and babies. And the reason for that is I wanted to take a comparative perspective. And so much of the animal behavior literature, particularly the primate literature, we have information on what males are doing with babies, but there's very little information on the relations between fathers and even men, brothers and so forth, and offspring as they mature. We just don't have that information. And so I made a decision, which was a good idea because the book was already too long. But you're right. And that's going to be another book someday, not by me though, by, I think Lee Gettler is actually looking at older life stages, and that will be his book perhaps later on. But I don't know of anyone who's really looked at what you want us to look at yet. It would be wonderful if we did. These are really great talking points for future research.
Thank you. Very, very quick interjection. This is one of the prerogatives I get by moderating. I think that what's happening here, Karsten, is that I always tell the students, we measure what we can, not what we should or we'd like to. And so it's so much easier to, I mean, the challenge has been, how do we measure investment, right? I mean, only because it's more complicated, it doesn't mean that it is not extremely important. And so we end up doing just proximity, caring, and contact, because those are easy to measure, not because we think are central to the story. Again, biases and doing what we can.
Karsten's question, though, reminds me of a subfield in my own field, which is, Karsten, have you followed the work on legacies and wills and what mothers and fathers are doing? They're kind of like dung beetles in that they actually, they're the only animal species I know of, maybe acorn woodpeckers, where you have kind of parental investment after death. And they go on investing after the parents themselves are dead and gone. And in humans, we have this. And there is a very rich literature on how fathers and mothers differ in the way they leave their assets when they die, studies of wills. And I talk about it a little bit in Father Time, starting with Charles Darwin's own will. But it doesn't really get at your question for other animals, unless Dr. Trumbo wants to talk about the philosophy of male and female care after death in dung beetles, which I'd love to hear.
Well, actually, I work on burying beetles. I don't know too much about dung beetles, but I was back to your point you made a few minutes ago about how fast some of these things can occur. I just point out there's a very nice set of studies by Rebecca Kilner, who also worked with burying beetles, on evolution of parental care and just removing parental care, and just, this is not looking at male and female care, but just having no care versus full care and seeing how fast things evolve. And she has about 40, 50 generations now and has seen changes in larval behavior, their begging behavior. The larvae are more cooperative when the parent is not there, when they don't have to compete to beg from the parent. There are morphological changes in the larvae to allow them to get at the resource easier since they don't have the parent to help them. And so, yeah, these things are happening pretty fast. I mean, their nesting behavior of the parents before the larvae ever arrive is different because they have to prepare the resource sort of more effectively, since they're not going to be there to maintain it after the larvae arrive. And it's just, if you're really interested in how fast, anyone's interested in how fast parental care can evolve, I just point out it's a remarkable set of studies that she's done in her lab at Cambridge.
I love that study, but for a different reason, because it describes what goes on in my own family, that my daughters, when I'm present, often aren't get in conflict with each other. And when I'm not there, they're just wonderful with each other. And it's just that study was so human. Who was the first author? It was Raybot or someone.
There's been a couple of different first authors, different postdocs in her lab. Yeah.
It's just a fabulous paper. I gave it to my daughter-in-law to read.
Thank you all. Sari has been very patient. Sari, it's your turn now.
I was actually going to go back. My questions search from the talk to the answer to Raga. And so we are going back to the owl monkeys and the stepfathers. And Eduardo, you're discussing that they are not overtly agonistic, but what about being affiliative? How fast do these stepfathers take over the role of as a stepfather? Are we talking about within days? And perhaps most probably there will be a difference between if there's still an infant around versus when it's only juveniles, right? But would you have data on that?
Yes, we have data. Do we have enough data, satisfying data? No, we don't. And this is something that Alba and I have been talking a lot and really hitting our heads against the wall, saying, how come, why is it that we have so little data on what males are doing? And I'm going to share again something about the natural history that I think has been very, very influencing on everything we've got and what we didn't get. we can, and this is, I mean, for this audience, I think it'll be interesting. We still cannot reliably identify individuals, not even males and females, unless we capture them. Nobody has succeeded in our project in 28 years to say, I can tell that individual from that one. I think that with Titi monkeys, from what Tony and others have told me, it is more likely. What this means is that the only way that we can be sure we have a replacement, I mean, it took us many years. Now we go to a group and we feel there's something in the air because we've been following, etc. Oh, gee, something must have happened. But then for us, we talked about having 10, 15 groups, 20 groups to collect data from. Which groups do you collect data from? You don't know on the go. I really think that what we should be doing the next year or so, I mean, now that we have the genetics, is finding ways where we can sex individuals right away on the field. And ideally, we can identify individuals in some way to say, my goodness, these are the 2, 3, 4 groups that we should focus on because we know now they have step parents. The truth is that it's taken us so long is because we needed to go back and look at the genetic samples we collected over 10, 15 years to start making sense of things that happened in the past. And so we all know how difficult it is to collect behavioral data. We're out there, okay, who do we go observe today? And you just don't know if you're definitely observing a biological father or not. Now, we published one little paper where we documented well the process by which, because we witnessed this one in detail, how the intruding male fought with the resident male and moved in. This was in December, and what that male did as it moved in, and within a few days or weeks, this new incoming male was providing some care and transporting the infant, but that infant did not survive. Now, no, we didn't get any explicit negative interactions between the male and the infant. So I think it's a horrible gap in the data to have. We have so little. We're hoping to go and in the 2013 paper, Children of Divorce, that Marian Hook wrote, she describes again some differences in terms of proximity and physical contact, but I'm sorry, I feel awful saying, but that's the truth. I don't feel that we have data that I feel comfortable making claims. Again, I feel like I cannot wait to see David's data from the lab, because going back to the comments by Alex and Sarah, we talk about all fathers being great. Well, I'm sure there has to be variation. And we do not have the resolution in the field to capture the specifics, the details of how these males vary in what they bring to the young. And I'm hoping that research from the lab, David, for example, has been doing a constant videotaping of the male, of the female. We're gonna start better the interactions between male and infant that are telling, that may help us then, may inform what we can go look for in the field, because we know it happens in the lab. So, you know these guys, sorry, you don't make it to Argentina, but you've seen the Titis. That's all I can say, really. Not a lot.
It's a shame, because it makes it so much more difficult, right? If you can't recognize your individuals and you're like blind kind of walking in this forest, and then like, oh, there is a monkey there. And then
the problem is that you have to commit ahead of time. So say you have a student who's going to study, David, take David. He went to Peru. The positive things. We were able, Marcelo Rotundo went there, boom, in 2 months, boom, you have radio colors in 7 groups of Titis, right? Bingo. Now you have 7 groups. You're starting. You don't know the relationship between the individuals. You're starting getting to know the individuals. And then who do you go study tomorrow? Who do you go study the following day? And it is a soap opera. And if you focus on 2, 3 groups that you get to know well in detail, you may never see a replacement there, right? Just pure chance. And then you don't get published because it's only 2 or 3 groups. And if you focus on 10 or 15 groups, then you start missing things. I don't know the solution.
But in other species, it's easier to recognize individuals. I think that we all agree that there are certain species that, with the owl monkeys, Titi monkeys being examples, that it's just extremely difficult to recognize individuals, while other species, it's easier, and therefore you have this information kind of instantly, right? That you have a change of individual, you have the sex of this individual, and so forth. So
I mean,
it's a
I always tell the story, the Titi monkey colony at Davis that has been going for 50 years. This is a captive colony of Titi monkeys. You're looking at the monkeys from a meter, right? In captivity, they have different tags for the males and the females. And these are not monkeys running wild 20 meters up in the forest. These are monkeys in a cage, and they tag them because it isn't easy for you to tell who's the male and the female. Tony, what you and others have told me, you guys have ended up watching the Titi monkeys at Tipuni so much more than I have. And I think that for the Titis, my impression is that all of you are settling in some consensus that you can tell the males from the females somewhat. What's the story with individuals?
Well, you can tell the males from the females somewhat if you get a really good look at them from close up. If you see them urinate, the urine stream takes a little bit of a different trajectory coming from a male versus a female. Individuals, I have for years, and you know this, said I'm not the best person at doing it, and other people are much more skilled. So some people have a much better eye for what details are individually distinctive and can do it, not just with the TDs and the Sakis, but with some of the other taxa that.
That's a lot of confusion there, because students will tell, I can tell that individual. They can tell that individual in the context of the group, right? But that's not enough. You need to be able to tell that individual, and when that individual now is not in this group, is in that group, right? And this is one of the things where we have all kinds of biases. We think we know and we don't.
No, but if you have an idea, it's like, oh, the individual has changed, like you said, that you tag all your individuals in the wild, otherwise you can't recognize them. So if there is a change in individuals, like let's say the male has disappeared and a new male has come in, it must be that you recognize that the tagged individual isn't there anymore, right? And you just have an untagged individual that you don't necessarily know if it's a male or a female, yet most likely will be the male, given that you lost the tagged male, right? So with that information, obviously you go ahead despite all the uncertainty of that, the idea of that individual, right?
Right, but we're not tagging all individuals. It is, I mean, you play your cards. I can make the claim, and in a primatology audience said, well, we've captured 300 animals. Their way of thinking about it is, yes, over 30 years and studying 10 groups. You do the math, and that means that we're capturing on average 1 individual per group per year out of the 4.
So how do you recognize the individuals within the group if there is only one tagged individual? Sorry.
I mean, over the course of 30 years, it fluctuates a lot. There are periods when we get to tag many individuals. And like you say, if you by size, by who is nursing, et cetera, but suddenly you may find yourself falling into circular reasoning, like we've talked about it with you, Sari. So it varies a lot. That's something we're trying to do now, actually, Alba and Jonathan, I mean, the new wave, finding ways where we can identify individuals from fecal samples much more readily, because the tagging, even when I think we've done extremely well for primate standards, it doesn't allow us to move to a different kind of phase where we can every season. I mean, think about what people do with the marmosets and tamarins. You set up traps, and every year, bingo, boom, they're trapping 30, 40 animals in 2 months. I don't think we can do that. I don't think it's reasonable to think we'll do that with all monkeys. So how do we kind of make an inflection point of really going to a different order, to scaling up, identifying? That's one of the challenges I think our younger colleagues have for the next decade. You have to scale up in a way where every season, bingo, we're going to focus on these 10 groups, and I know all 20 adults from the go. that's things we're pondering and exploring how to do it with doing a quick identity analysis from fecal samples, because the capturing with rifle, we capture the owl monkeys with a rifle. I mean, the same way that we've done it with Tony in Tiputini. Tony has studied many primates of all species, but you cannot scale that up to the level where we were. I mean, yeah, Sarah. Sorry, hold on, Sarah, bear with me, because I want to be, Sarah, if it is quick. If not, I'd like to give a chance to Adriana or Tomislav who haven't asked, and then I'll come back to you.
Really quick.
Yes.
Eduardo, how do you think the monkeys are recognizing each other's smell or noises?
Research in the lab by Andreas Penseisenberg, doing some bioassays and looking at olfaction, and she did some testing of olfaction. We know, at least in the lab with true experiments, the animals can recognize sex, can recognize age, etc. The animals are, we just published a paper Alva did this year in AJP describing olfaction in the wild owl monkeys. The animals are marking a lot. The animals are moving through the forest, lowering their rump like the Titis do, rubbing their glands against all kinds of substrates. David tells me the same. So, and there's a huge vocal repertoire. We were finally able to describe these very loud calls that the owl monkeys made. I mean, with playback experiments in the field, telling that there's a female call, there's a male call. We can do playbacks, and depending which call we play, we get more or less females. So vocal communication, olfactory communication, those are, I'm sure they are playing a big role. Mm-hmm. Adriana, and then Tomislav.
Hi, everyone. Hi. Thank you, Sarah and Eduardo, for a great talk. I think I'm just going to take you off a little bit of the discussion that we're having right now. My question goes back to one of your first slides, Eduardo, but this is open to both of you. So we know pair bonding is one aspect of the social system, and then we have parental care. But to what extent does pair bonding defines who does biparental care in like monogamous species? And then I'm introducing a third component of the system. So do you see in an evolutionary perspective, if pair bonding is somehow linked to parental care in different species? Is it a prerequisite, or?
I definitely want to tap on, I mean, we have so many colleagues in the audience that I'm sure can help us get a richer answer. In primatology, we need to go back 30 years, and Robin Dunbar was already doing some analysis postulating that you need to have pair bonding to get then paternal care. Maren did a review of the mammal literature for the 2014 paper in Proceedings, where we described genetic monogamy, and we looked at the association between pair bonding and whether you have what we called close or relaxed proximity. I mean, the kind of coordination you need between male and female, and again, the extent to which they're more or less likely to be associated with paternal care. And for that analysis, I think it was about 16 socially monogamous mammal species that she reviewed. There seems to be an association between pair bonding is more likely to occur to paternal care. Tim has left, but he also speaks of this in the Mammal Society's book. It is less frequent that you do get paternal care in the absence of pair bonding. But I really would like to hear also, I mean, Karsten has really done a lot of reviewing the mammal literature more broadly than I have. Now, if we're trying to connect this with some of Sarah's thoughts and Alex, because of his expertise with the balls and neurobiology, we really, I mean, for making progress on this, we'll have to get much more sophisticated than just talking about pair bonds, right? What is the pair bond? Because now suddenly, and I don't really get very excited with people talking about bonds because two animals are in proximity, right? For me, there has to be something more than that. If we're hoping that we'll identify some kind of proximate mechanism, the way that Sarah was describing, where you seek closeness. But it's not closeness that you care about, it's what the closeness brings. If the closeness brings physical contact and touch, and that elicits some kind of dopaminergic reaction, well, then it's really that what's going to play out in your brain. It isn't the closeness per se. I always make the joke that, I mean, if we're going to go by closeness, I have a remarkable pair bonding with a bus driver every morning. I mean, cram bus, right, really. So I think that you look at the literature, I think that it is so much more likely that if you have pair bonding, you have engagement of the male in the care of the young, that the other way around. Pair bonding is not enough to elicit male care, but it seems to be, in most cases, I mean, you can even go back to Clayman's 1977 and 1981. It seems to be like most of the times you have the male engagement, you have a bond. Now, what do we mean by bond? I mean, with the Titi monkeys in captivity, I mean, when my colleagues from Bill Mason to Sally Mendoza to Karen talk about a bond, is because you really have an emotional change. I mean, there's a disruption in the emotional stability of the pair when you break that relationship. For me, a bond, you really start getting to something when you show that, I mean, how strong is a rope? Well, tell me how much force do you have to apply to break it, and when it happens, when it breaks. And with the Titi monkeys, you make separation studies, you look at cortisol, et cetera, you give them options, and the partners will consistently choose their partner over a novelty. That's unusual in mammals, right? Most males will go for the novelty, not in Titi monkeys, in captivity. In the field, we have no idea. We have no data. The closest we've got is that when we capture, going back to captures, I wish I had known we would capture 300 animals, because capturing one of the partners is a separation experiment of sorts. Only that we never thought we would be doing 300 of those. And consistently, the remaining partner gives calls of contact. The remaining partner is showing some indication of distress because now you've removed the female or male. But who else would like, I mean, can somebody help me, Karsten? Yes.
Yeah, so I personally do not believe that paternal care needs pair bonding. I think both of them might often evolve under similar ecological conditions, but I don't think one is the prerequisite of the others. My favorite example is the art wolf, where the male and female don't really like each other, but the male still comes every night to spend half the night with the pups to protect them, and with this he has 5 times higher reproductive success. And this is why paternal care evolves. It evolves because it increases male parental care, not because the male likes the female and he has to help the female. And if I would make a guess, when does pair bonding evolve? It's like what Eduardo says, it's often physical closeness. And most mammals are small. People forget this because people mainly study large mammals, but most mammals are small. They are endotherms. They spend a lot of energy keeping warm. And social huddling is one of the main mechanisms to reduce these energetic costs. Now, males hate males, females hate other females. If you want to save energetic costs and huddle with someone, you need to huddle with an opposite sex partner. And I would guess this is the start of pair bonding in many species, because when you like someone and you're a small prosimians, you're a small New World monkey, you're a small mammal or mouse, and you cuddle together with somebody for half of the day, you save a hell lot of energy.
But I agree with you, Kirsten, but my understanding was, I'm trying to remember some of the somebody help me out if you have better background on neurobiology than I do. I mean, one of the arguments was that liking the female, now I'm not talking about liking the female, but if you get some kind of reward system in the brain activated, that
Yes, but I talked about evolution.
I remember,
I didn't talk about, I talked about evolution. I didn't talk about mechanisms. So this is different aspects.
Right, but you're saying that the male doesn't go back. I really like your argument around that you may only need another warm body for you to seek proximity. But if that proximity ends up being with another individual with which you end up reproducing, that's going to be very different. I mean, if you have a Y maze and you can seek proximity, you're a male, and you present them with a Y maze, and just randomly flip a coin. You may either go address your thermal challenges with that one who is the same sex you are, or with a female. One is a dead end in terms of you, in terms of this seeking of huddling being passed. I mean, nothing's going. You're still going to be warm, but you're not going to reproduce. Imagine a male that every single night for the whole mating season seeks heating from another male.
Yeah, I mean, that's my point. That's why it's much more likely that pairs of one male, one female evolved than of two females or two males.
Again, please.
That's exactly the point. That's the reason why, if there's an evolutionary trajectory to save energy by huddling, it's most likely that it will be a male and a female pair and not two males and not two females.
But I think that maybe it comes down to how we're thinking of pair bonding. I don't think about pair bonding as I see it as it can be a very, very basic mechanism of seeking proximity. And why proximity? Maybe because of thermoregulatory challenges, for example. I think it's fascinating. You're right in saying that those of us studying primates forget about the rodents, and then so yeah, on that we fully agree. Tomislav, maybe he had to leave. Oh no, there you are. You're muted. You're muted, Tomislav.
Can you hear me?
Now we can.
Good. Thank you. Thank you for a very interesting discussion. Actually, I came to a thought because you took genetic samples of your individuals. We know from Catholic Church that celibacy in priests is explained in behavioral ecology, human behavioral ecology, as a kind of a kin selection. Have you tested regarding stepfathers how much kin selection exists in your troops of individuals?
No, we have not, but we have the data. We just haven't been able to get to it. It's a fascinating. We have not really started to examine the genetic structure of our population to see the extent to which. So I talk about groups. Each group occupies anywhere between 5 and 10 hectares. And so you can think of it as a neighborhood. But how that neighborhood is structured may be around kin, where the animals moving in and out are not all evenly related. And I mean, is it my uncle who's moving?
I think it will be very interesting to test.
Yeah, absolutely. Absolutely.
Yeah.
No, it's something pending. Guys, it's been such a lovely discussion that I didn't realize we're past the hour. So let me first stop by, usually what we do is we thank our colleagues who may have watched the fine seminar in YouTube. So thank you for doing it. We encourage you to also join us via Zoom if that's possible to you. With that, I'm going to stop.